Good morning, folks. Anybody there? Can you type in or whatever? I can't hear you speak, but I can speak to you. Got to take roll, so let me know who's there. So far, we got 3 people online, but our clocks might be a few minutes off, so we'll give them a couple more minutes to register or show their presence. We're going to be on page 562, I believe. I think the last thing we covered was on page 556 was rheumatoid arthritis. And one of you guys want to confirm that for me? We haven't covered it. I do want to cover it. Maybe rheumatic fever and rheumatoid arthritis. See if that's the last thing we covered. Give me a yay or nay with that. So Destiny, your yes means that I've covered rheumatic fever and rheumatoid arthritis. Is that correct? Sure is a cumbersome way of doing things, isn't it? Okay. Ah, good to see all you guys coming on. Okay, so we evidently covered transplants also. How we had to match up the antigens that created the worst rejection reactions. So let's go over to page 562. And you see in that second column, it says immunodeficiency diseases. Looks like our time lapse is a little bit slower, so may come across looking kind of weird in terms of talking. But as you look at that paragraph, immunodeficiency diseases, you see there are 2 categories. If you look in that paragraph, you see 2 bold print terms. primary immunodeficiencies, and secondary immunodeficiencies. So what you want to know about those two is you've got a great definition for them in this case. You see the primary immunodeficiencies are caused by a genetic defect. And as you read through there, you can see there's a problem with the T cells and the B cells that we talked about before the spring break. So secondary immunodeficiencies, as you can see down there, they give you several problems that can bring those about. Infectious agents, and one we'll cover is going to be the HIV that can ruin a person's immune system, and then two, malignancies, and also immunosuppressants. You know, one of the things that people don't realize when they get a transplant, in most cases, they have to take some immunosuppressant. Hopefully it's not a lot, but anyway, it does affect your immune system. Some of those drugs you see on commercials on the television, and they've got the ending of the name in parenthesis says MAB, monoclonal antibodies. Those are meant to suppress your immune system to some degree so that you can have some relief in our talk about rheumatoid arthritis, but also in the sense of being able to keep a transplant. So those are the two categories that you want to be familiar with. Now, if you look over on page 563, let me check again, make sure that some of y'all are here. Okay. So as you look on that page 563, some of you see that little application box about the boy in the bubble. Some of you may have seen that movie. I didn't get to see that, but that little fella had a severe immunodeficiency. We're not going to get into that completely yet, but look down underneath that applications box and you see primary immunodeficiencies. Now, look at that bold print term, if you would. I'm going to get you to do a little bit of thinking in terms of this and apply what you knew in anatomy and what we've already talked about so far in this course. You see that big word, agammaglobulinemia. Now think about that for just a minute. That's probably a word you have not seen in your life at this point, but break the word apart. Think about how back in anatomy in Bio 210, when you had your vocabulary test, how can you break that up? Can you see ways to do that? Take just about 15, 20 seconds and see if you can recognize terms in there that are put together to describe a condition. Great. So as you look at that, if you're just working from left going to the right, when you see A in front of a term or a condition, as you see I on the end over there to the right is a condition, then A means without. And gamma globulin is another word for antibody, isn't it? Or you could use the word immunoglobulin. And then you see the ending, the suffix, emia, and that means a lack of something in your bloodstream. So if you have anemia, then you lack red blood cells in your bloodstream. So working still in terms of a little bit of thinking, when they take an assay of your blood and they check how many antibodies you have against a particular organism, you may come up with none. So in that sense, you sort of have A-gamma globulinemia with that particular antibody. But in this case, the person does not have any antibodies. So think about this. They draw your blood, they draw that child's blood, and they don't find any antibodies in there. And that means a particular cell is either absent or has a problem making those antibodies. What cell would that be? And that would be a B cell. B cells are the ones that produce the antibodies for us. So knowing that the antibody is produced by a B cell, if you don't have antibodies, then you've got a problem with your B cells. Now, what could be the problem with the B cell? Think about that for a minute. What could be non-functional or dysfunctional in that cell so that it cannot make an antibody? Take a few seconds to think about that. Okay. So think about it. What process produces antibodies? Now, you jump back from that question, you think about, well, what are antibodies? In which biological category do they fit? You only got 4 choices, right? You have proteins, you have carbohydrates, you have lipids, and you have nucleic acids. So hopefully all of you are saying those are proteins, therefore there's a problem with protein synthesis. So what are the steps in protein synthesis? You learned back in anatomy and physiology, there were 2 steps in terms of protein synthesis. So you go to something like that and you say, well, let's see if transcription is actually occurring. And if transcription is not occurring, then there's where you got a problem for whatever reason. You have to try and solve it at that level. If you find out, well, transcription is working, in other words, a messenger RNA molecule has been made, sent out into the cytoplasm, but we don't end up with the product that we need in terms of an antibody to protect us. So therefore, translation has a problem. Now, well, you got 3 components that are involved in translation, right? You got the messenger RNA, you got the tRNA, and you got the ribosomal RNA. So somewhere you've got a problem with one of those 3, or maybe a couple of them. You know, the transfer RNA is made in the nucleolus and so forth. Just trying to help you to put together a thought process that you could work your way through if you were involved in trying to determine why this B cell doesn't produce antibodies. A little bit of thinking there, taking what you've known in the past and applying it presently to this situation. One of the ladies that I had a number of years ago had a son who did not produce antibodies. So what she had to do was bring him into the hospital, I guess a physician's clinic or whatever, and they would infuse into that young fellow's bloodstream a solution of antibodies. Now, Where do you get antibodies? Well, one of the things that we can do today with a pint of blood is we can separate the components of the blood. And in doing so, we can give somebody packed cells, we can give somebody platelets and so forth, and we can also get antibodies from their serum. You'd have to let the blood clot, then get the serum, and in the serum you would find those antibodies. There may be a business at this point in time where people are taking, if you remember how we talked about monoclonal antibodies, they're taking cancer cells, they're fusing them with B cells, specific B cells that would produce, say, an IgA and IgG against cold viruses. And so they've got a little industry going. Therefore, they harvest those antibodies that the cancer cell, remember the hybridoma, the cancer cell and the B cell that's making a particular antibody. And so they've got a little factory there. And they take those antibodies and then they mix them all up and they sell it as a product that can be infused into a person's bloodstream and gives them somewhere between 5 and 8 weeks worth of protection. So they don't have as many colds as they used to and can fight those off and have some semblance of normality in life. Everybody okay with that? Any questions about it? Okay, nobody's responding with a question, so that's okay. Now come down, if you would, under to the next bold print term, and you see the heading Digeorge Syndrome. Now, you look at that, and nobody knows what that is for the first time they look at it. It's just named for a person because of that person's contribution in terms of understanding this problem. But you see here we have a deficiency of T cells. So think about the function of T cells. We talked about helper T cells. We talked about suppressor T cells. We talked about cytotoxic T cells. So you can see that any of those that are missing, you got a problem, especially where your helper T cells. What do we call the helper T cells? We call them the commander-in-chief. So if those are diminished in number or absent completely because of a genetic problem, you got a problem. Any questions about the Digeorge syndrome? I know I keep looking up because I've got 2 monitors here. Got a little surface, and then they got the big screen. Anyway, let's go to the second column, and let's look at severe combined immunodeficiency. And this is the situation in which both B cells and T cells are absent. That's where the boy in the bubble comes in. He had a genetic problem. You don't have to, you can read through that first paragraph, but I'm not going to hold you responsible for a defective gene for IL-2, which means interleukin number 2. But anyway, he had a problem. We know he died, I think, around 12 to 13, 14 years of age. Now, if you'll look down in that category under severe combined immunodeficiency, come down 1, 2, 3 paragraphs, and you see this italicized word, gene therapy. So I want you to look at that, read that. Come down about the 4th line there. You see it says a few children have had their bone marrow cells removed. So you got to do a puncture there, sternum, iliac, or something like that. And it says those cells that they have pulled out have been infected with a reproductively deficient retrovirus carrying a missing gene. Now, we're not going to get into the gene and all that. It's real interesting stuff. And it's amazing what they're doing. Now, think about a retrovirus. That's a category of virus that you know. You go back to chapter 10 and remember those pages that we looked at. You had the RNA viruses, and then you had the DNA viruses. And so a retrovirus is a type of RNA virus. Matter of fact, the HIV is a retrovirus. So they have studied that virus, and they know how to stop the replication process. But they don't destroy the virus's ability to get into one of those cells that came from the bone marrow. So that reproductively deficient retrovirus can enter one of those white blood cells, a stem cell, like a hematopoietic stem cell. And it's like a little truck. It gets into the cell and drops off the gene, the correct copy of the gene. And amazingly, the cell knows exactly what to do with that, incorporates it into the DNA, and now that gene is working, and it begins to produce the components that are necessary for an effective immune system. That is amazing. Now, we spent millions of dollars, paid a lot of PhDs in immunology to study that and discern how we could change the gene in the cell. And so once that cell starts working, once that gene starts working, that kid's immune system is up and running. That's why we spend millions of dollars to see if we can help people. Fascinating stuff, just fascinating. Do any of you have any questions about primary immunodeficiency diseases? Okay, so we have no questions. So you look down at the bottom of page 563, and you see secondary immunodeficiency diseases. You can see these are acquired sometime during the lifetime of the person. And as you look through there, they'll mention things like autoimmune diseases, and that's what we've got going on in this country to some degree. Let's see. Amanda has a statement here. Let's see. I saw a show where they are turning the gene for infant red blood cell production to cure sickle cell anemia. Okay, that's wonderful. And that's why we study this stuff, to try and help kids. But boy, you're getting down to just the molecules, aren't you? That's fascinating that we can do that. Marcus, and you're asking the question, so a virus can change our DNA. Well, of course, in the treatment we were talking about, we took out the genetic material that would, or we compromised the genetic material of the virus that would cause it to replicate and create a problem. So somehow they have either removed that, or they have made it defective so it will not work, and they put the correct gene in there. So when the virus gets inside the cell, then that copy of the gene can now be assimilated. Can viral DNA be assimilated into human DNA? The answer, I believe, is yes, it can. And that may be one of the ways, Marcus, when we get cancer, we think of cancer as maybe caused by smoking or some chemical like, what was it, Roundup? They're having advertisements on television about getting some money back if you develop, I think it was Hodgkin's lymphoma or something like that, from working with that chemical product. So yes, a virus can get into your cell and turn it into malignancy. think about chapter 10 and go back to transformation. We talked about the cytopathic effects of a virus, and one of those possible problems could be transformation. So yes, a virus can get inside our cell and incorporate itself into our DNA, and then we got a big problem in some cases. Okay, good question. Now let's look on page 564, and we want to look at one type of secondary immunodeficiency. And you see it on 65, 64, come down to acquired immune deficiency syndrome. Now we shorten that up to AIDS, don't we? So you want to know what AIDS stands for, acquired immune deficiency syndrome. And if you look, as you look into the first 4 lines on that paragraph, you see the bold print term for the causative agent, the etiologic agent, and that is the human immunodeficiency virus. Now, you guys know how that is picked up. We can get it through doing drugs, as they talk about on the street drugs that are sold, and people want to get high on them, and they're very careless about sterility and so forth. So they can be picked up that way. They can be picked up through a blood transfusion. As a matter of fact, I think we, years ago, and probably 30 years ago, a couple of the guys that worked here at Tech had some surgery done and got a transfer, they received a pint of blood from someone, and in that blood was the HIV virus. So evidently, if it wasn't screened, or if it was screened, it was done so inadequately, and that person ended up developing AIDS and dying from it. So that's another way. And of course, you can pick it up sexually also. Now, when a person tests positive for HIV, that's what they label them, HIV positive. And we've got some drugs at this point. We're not going to get into all of that, but some of you may, as in your professional careers, get into it. But we have some drugs that will slow down the replication process of the virus. Now, what are the side effects of that? I have not looked that up, but it seems to extend life. Now, there's another possibility. It's called TNT. And most of us, when we think of TNT, we think of an explosion. Some of us used to shoot firecrackers, and they were labeled TNT. And they really weren't, because you can really get blown up with TNT. But anyway, the TNT in this case stands for triple nutritional therapy. Triple nutritional therapy. So I've got a little note here. It says, has been found to drastically reduce the decline in the immune response and therefore decrease the development of AIDS and death. So you can look that up and find out what it is. It's like a mega shot of various sorts of substances: selenium, vitamin E, vitamin C, the groups of vitamin B, different kinds of B6, B12, and so forth. And so that seems to prolong the life of the individual and give them a quality of life where they can actually continue to work. You don't have to be scared of getting AIDS if somebody is working beside you. So like the coronavirus, which is transmitted through the air into people's respiratory tracts. But you can work with someone and you're not going to pick up AIDS from them unless you engage in those behaviors that will allow transmission to occur. So on page 565, when a person develops what they call AIDS, that means their T cell count has diminished to a certain point. I want to say it has to do with the number of T cells, the helper T cells that are in a person's bloodstream. And they can tell the difference evidently from the cytotoxic T cell and the suppressor T cell and the helper T cell and so forth. So when that number reaches, instead of saying having 600 per milliliter of blood, you're down to 200 or 150, that's when they declare that you have AIDS. So it has to do with the number of T cells, probably helper T cells, that are present in that person's bloodstream. Then they're called immunocompromised or immunodeficient. They've got what they call AIDS. When that happens, as you look on page 565, you look at table 18.7, and you see it says infections frequently found in AIDS patients. That's because their immune system is compromised and they get these diseases, these infections that you and I, who have a good immune system, don't see. Now, there are a number of them, but we're going to check off just a few of them. So if you would, as you look under bacteria, table 18.7, and you come down under bacteria and you see Mycobacterium avium intracellulare. Now look at that word avium. Does anybody recognize another word that is similar to avium, or do you know what that word means? Jamisha, I see that you have sent in a thought there. And droplets, one of the things that goes on when we cough and breathe is we have little mucus droplets come out of our respiratory tract. So my understanding is that that's why they want you to have that, what do they call it, social distance, an acceptable social distance. So those little droplets that are in the air that you would normally inhale through your mouth or your nose do not get to you. I think, what is that, about 10 feet now? We're going to have to see if we can work something out about that in terms of lab. So maybe we have half the people come to lab and they're 10 feet apart. I don't know how we're going to work that out. But anyway, saying it's not airborne, I'm not sure about that, because how else do you pick it up but the droplets through the air? And if I'm wrong, that's okay. I've been wrong a number of times in my life, and I probably will be again. But that's my take on it, Jamisha. Thanks for that question. Okay, Eugene says an aviary, and he also says birds. That's right. An aviator is a person who flies an airplane. So this gets picked up through the air. Now, this is present in birds. You and I come in contact with birds every day. They're out in the environment. Some of you have bird feeders, and you like to see the birds come. You like to be close to them and look at them, maybe through binoculars or whatever, or just have them around. You like their songs. And some people have pet birds, don't they? I had a student whose mother had cancer. And so her mother was getting treated for cancer. And then she began to develop some signs and symptoms, respiratory problems, and so forth. So she goes into the doctor, and her daughter, who helped her get there, was an LPN. She was becoming an RN. And so they diagnosed her as having Mycobacterium avium intracellulare disseminated tuberculosis. And so the doctor asked her, says, this is usually picked up with birds. Do you have birds? She says, yes, I have 9 parakeets. Get rid of them. That's your problem. Now, you see, her mother was already compromised, right, with the cancer and the treatment of the cancer. So that compromised her. and allowed what you and I can come in contact with and not have any problem at all with, they set up an infection in her respiratory tract. Got rid of the birds, treated her for that condition, and that was the end of the problem. Everybody okay with Mycobacterium intracellularly? I had a friend who got a liver transplant out in Nebraska. He developed it. And so I think he had a liver transplant. And of course, they put him on immunosuppressants. Well, he developed Mycobacterium or disseminated tuberculosis while he was in the hospital. As soon as they backed off on the immunosuppressant so he didn't reject the liver, his immune system rose and knocked out the Mycobacterium, like you and I do. We don't have any problem with it because our immune system is in good shape. Okay, let's go to viruses. Same table, 18.7. You see herpes simplex. Now, when you think of herpes simplex, most people think of fever blisters. Okay, and that can be a case. You see it says skin and mucous membranes. And then people, of course, can, if they're immunocompromised, that little sore, herpetic sore that is so painful for most folks, that can spread into the mouth, go down the throat. It can actually get up into the respiratory tract. We don't realize it, and it doesn't happen very often, but these little herpes viruses can actually take you out. We look at them as an inconvenience in most cases in terms of fever blisters. So that's a viral infection that could show up in a person who has AIDS. Any question about the herpes simplex? Okay, let's look down at fungi. And I want you to check off the first one, Pneumocystis. And you see the species there, Gyrovecki. And you look over to the right, it says Pneumocystis carinii pneumonia. They used to call it Pneumocystis carinii. And why they changed it, I'm not sure. But it is a fungus which you and I carry in our respiratory tract. All of us have it. As soon as we were born, this little guy starts colonizing our lungs. Now, obviously, he doesn't create a problem. But when your immune system is compromised, this little fella overgrows and often takes out the AIDS patient. They die of a pneumonia, and it's this pneumocystis that's causing the pneumonia. My father had cancer back in 1992 and died from it. Became very weak and spread to his brain and so forth. He never got pneumonia because his immune system was still good enough, still strong enough to prevent the Pneumocystis gyrovecki from overgrowing and taking over his lungs. So if you have a patient that has pneumocystis pneumonia, then that patient has a severely compromised immune system. You're not going to catch pneumocystis pneumonia from that patient. That patient's immune system is the fault. Do you have any questions about those three infections, one bacterium, one viral, and one fungal? Any question about that? So if you have no questions, then let's scoot over to chapter 19. And that's on page 585. Eugene, I see your name's up here with a question mark. Did you want to elaborate on that question mark before we move into the diseases of the skin and the eyes? Okay, that's good. Thanks. So as you look on page 585, one of the things you see is this picture of this older fellow, and you see all the lesions on his scalp and on his nose, on the side of his face. That's shingles. Now, typically they show it on the television as coming around the torso, don't they? Around the thorax or the abdominal region, the lumbar region. But it can go down the back of your leg, down your buttock, and down the backside, the dorsal side of your leg. But you see, it can also show up here. Shingles can be a very serious situation. Now, let's look on page 587. We're into the diseases of the skin. And since we're starting looking at these systems, one of the things we'll do is we will, okay, Shavia, great, I'm glad you're here. What we'll do is we'll review a little bit of our non-specific defense mechanisms that keep us healthy. So when we think about non-specific defenses for your skin, and they're actually including the eyes here. As you look on page 587, you see the lacrimal gland produces tears. And what is found in tears? Well, you got salt, don't you? Yep. You also have that, what was that enzyme? Anybody remember that enzyme? Contains that, is found in tears and saliva and mucus. Called lysozyme, okay? So that's a means of protecting our, in this case, eyes. And you also have, as you look down at the bottom of page 587, you have normal microflora of the skin. So we're learning that right, good, Shavia, good, Destiny, that's great. Glad to see that. So you see how we're actually bringing up from, what was it, chapter 16 into chapter 19. That's going to continue through the semester. So you don't want to just leave out reviewing old test material. You know, we talked about capsules on the first test. And here we are getting into these diseases, and we'll talk about capsules again. So keep all of that current so that you can weave it right into the answers of the questions I'm going to ask. So the normal flora help protect us. We want to reduce the number. We can't get rid of all of them. Of course, we're being told to wash our hands well and keep them away from our face. But there are good guys on there that actually seem to inhibit the growth of pathogens. So we want to get rid of them. We want to understand them and then use them as they're intended. As you look on page 588, you look in the first column and you come down to the second paragraph and you see sweat. Got salt in it. Staphylococci like that material. So they live well on our skin and we're never going to get rid of them. Anyway, that's one that can actually dehydrate some other bacteria, but staff have some techniques and abilities that allow them to grow in that salt. Okay, let's see now. So let's go over to page 589, and we're going to look at the heading called Diseases of the Skin, Bacterial Skin Diseases. We'll look at bacterial, we'll look at viral, we'll look at fungal. Okay, so we'll have three categories in terms of skin infections. So as you come down to bacterial skin infections, you see staphylococcal infections. And you come down about 3 lines and you see Staphylococcus aureus. If I remember correctly, I don't believe any of you guys carried that in your NAIR. I don't think you did. We'll have a lab at 10 o'clock, and so I'm not sure how that's going to go over. I've never done a lab on the on this YouTube thing. But anyway, we do want to know about Staphylococcus aureus. I carry it in my respiratory tract. Hasn't bothered me for 20 or 30 years. Evidently, my immune system's in pretty good shape. So, but you know, when I have a cut or something, I'll be aware of it. And the strain I have is MRSA, and so that could be a problem. So I've got a cut or something. Well, I remember, you know, I've got to wash my hands and treat that very carefully, and then have eliminated any problems that I think I might have had. Anyway, Staphylococcus aureus, gram-positive coccus. And if you remember in the lab, Staphylococcus aureus is not only catalase positive, but coagulase positive. I believe we did that particular test and showed the difference between Staphylococcus aureus and Staphylococcus epidermidis. Both of them create a problem, but Staphylococcus epidermidis is a lot easier to cure with an antibiotic. Aureus has these plasmids that tend to produce proteins that break down the antibiotics, and therefore it's called an antibiotic-resistant organism. Now, in terms of staphylococcal infections, as you come on down, you see a number of bold print terms. We're not going to do all of them, but you want to check off folliculitis, remember, that's an infection of a hair follicle. And then you come down. Any of you guys ever had a sty? Anybody had one? Yes. I had a couple of them when I was a kid. Very inconvenient. That swelling gets in the way of your vision. But anyway, a sty is a staphylococcal infection. Furuncle. I've always wondered where they get that name, furuncle. But anyway, that's the same thing as a boil. You want to be careful with those. Oh, Jacqueline, and Eugene, you've had styes, huh? Okay. We usually overcome them, but they certainly are inconvenient and uncomfortable. I think I have mentioned this to you, but when you have a boil, one of the things if you find it early, you know your body, as you take a shower or whatever, and you find there's this enlargement of some degree, and that's when you want to treat it when it's small. That's where you can take that hot washcloth, as hot as you can stand it, and put it on the boil for 2.5 minutes. That's all you got to do. 2.5 in the morning, 2.5 in the evening. And that heat goes into that area and it denatures the proteins of the bacterium. I think I mentioned Zeno, Z-E-N-O. That was a tool that I don't know where it was invented, about that big, like the little collapsible cell phones that we used to use. It would have a tip on it, and it would generate 119 degrees Fahrenheit. And you would put that on there You let it get to 119, you put it on, and you've got a little timer in that tool. And for 2.5 minutes, you let it sit there, and 119 degrees goes in there, denatures that protein. You do that in the morning, you do it in the evening. And I was amazed at how effective it was, and avoided it, because sometimes I'll have a boil want to come up and catch it early, and that heat really is an ally for us. You don't want to squeeze too hard on a boil if you're trying to get pus out of it. You don't want that sac inside there to burst. If that bursts inside, you got a big problem coming up, because now you got staph going out under the tissues. That could turn into necrotizing fasciitis. It could get into the bloodstream, and you'll have staphylococcal septicemia. Tough situation there. So you got to be careful with that. And then the last staphylococcal infection I want you to know about is at the top of the next column, and that's carbuncle. You see it says it's a massive lesion. It's actually a number of boils that are present, all in one little area. They usually have to lance those, drain them. I don't know if any of you have ever had a boil and had it open, and they pack it with maybe iodoform gauze. There's something else they pack it with, and I can't remember what the name of it is at this particular point. But the iodine does a good job. But then what they send you home with is they tell you got to change that maybe every day or 2 days. You got to pull that out of that hole, and then you got to pack it back in. Any of you guys ever done that? Not a pleasant situation. I've got a friend over in Darlington who has had that, and not fun at all. Let's go over to page 590. Page 590. We're going to leave the staphylococcal infections, carbuncle, boil, folliculitis, and so forth. I want you to come down to the bottom of page 590, and you see streptococcal infections. Now, as you look in there, as you look in that heading, you see scarlet fever, and that's caused by, you see, Streptococcus pyogenes. Now, if you remember, when we looked at those blood plates, and I'm going to try to show them to you again. I'm going to take my laptop down there, and hopefully I can stir some cobwebs up there so you'll feel a little more comfortable with the plates. That Streptococcus pyogenes was a gram-positive coccus in chains, and it was beta hemolytic. So as you read into this, you see scarlet fever is associated with a strep throat. But look at the third line, right in the middle of the sentence, right in the middle of left to right. You see it says strains of the organism that cause scarlet fever have been infected by a temperate phage. What does that mean? Infected with a temperate phage. What does that mean? That's the same thing as virally converted. You see how chapter 10 is going to come right back up in here and influence. That's right, Eugene. The virus has been incorporated. The DNA of the virus has been incorporated into the bacterial DNA. So it's that toxin. You see, it says enables them to produce that toxin, erythrogenic toxin. The toxin doesn't really hurt you, but it does create a rash-looking appearance. So That's why they call it scarlet fever. Now, you will produce, if you get scarlet fever, you will produce an antibody against the erythrogenic toxin. It's a protein. So if you get the strain that causes scarlet fever again, you get a strep throat, you're not going to have that change usually in your body because you've got antibodies that would neutralize the toxin so you don't develop the rash. So that's one infection that's caused by strep that I want you to know. I want you to look right above the streptococcal infections. You see that foot that looks so bad? If you don't have the color version of this, then you won't pick up on a lot of things, but you can see it's pretty splotchy. Now go up to the top where you see flesh-eating bacteria. Let's look into this little news article here. Come down to the second line, and you see it says newspapers and television sounded alerts after people began to die as a strain of Group A Streptococcus. What does Group A mean? Well, Group A means that it's got a particular antigen on its cell wall. As a matter of fact, we might be able to do that today in lab, where I show you how they can tell if they got that antigen. But that's an identifying factor. You remember Rebecca Lansfield studied these organisms and helped arrange them in groups. Now they call it M1 T1 Strep. You do not have to know that. Just know that in this case of the flesh-eating bacterium, it is a strain of Streptococcus pyogenes. A strain. That's all you got to know about it. Now, notice what it does. It's got the ability to eat away at the flesh of the patient at the rate of several inches per hour. So it's literally digesting you. It's eating you. And you don't do anything about it. You better plan your funeral. It's going to take you out. Now, what's creating the problem? Come down to the second paragraph where you see Australian investigators have recently found that this fierce virulence, there's that word that we talked about, right? Fierce virulence is due to a gene brought in by a bacteriophage that infected the strep bacteria. It didn't kill the strep, but it did enhance its ability to kill you and me. So this M1T1, and you don't have to know that name, you just know it's a strain. That strain comes about by being virally converted. It's interesting. You don't have to know the rest of it, but it's interesting just for your own knowledge if you want to read about how it digests the proteins of cells and tissues. Okay? So there's your second streptococcal infection. Look over on page 591. And you see in the first column, oh, Marcus, I see you had to put in iodine strips in your when you had that boil. Very uncomfortable, wasn't it? Sorry you had to go through that. As you look on page 591, you see Pyoderma and Impetigo. What I want you to do is highlight Impetigo. And you can see it about 4 lines down, 3 lines down. Highly contagious Pyoderma. And pyo, of course, is a root word for pus. Derma is your skin. And you see it says it can be caused by Staphylococcus and Streptococcus, or both. A nice little picture down at the bottom of the page. You can see the lesions around the child's nose and mouth. And children are the ones that typically get it. Adults can, but the kids have a hard time keeping their hands off of it, and then they spread it around. So it is very contagious. Nobody dies from it, but it's very easily transmitted. Okay, so you got 3 strep infections, right? Several staph infections, 3 strep infections. Any questions so far about those bacteria? Eugene, your question about is that also called hand, foot, and mouth disease? It's not the same thing because this is a bacterial infection. Hand, foot, and mouth disease is a viral infection. You wouldn't have any pus, you'd have some lesions, but you wouldn't have any pus. Okay? Bacteria produce the pus, or bacterial infections are noted for their pus, but this one's a bacterial infection as opposed to a viral infection. Okay? Still on page 591. You look in the second column and you see, come about halfway down, burn infections. So you look at burn infections, you come down about 4 lines. Anytime you got a burn, there's this awful possibility of having this organism. You see the italicized genus and species? 80%. Pseudomonas Aeruginosa. I was looking up there for a second. Sorry about that, guys. But I want you to highlight Pseudomonas as a primary cause or prime cause of life-threatening burn infections. Now, you've been exposed, I think, to Pseudomonas. If I remember correctly, you had the tryptic soy agar, and you had sort of a greenish blue pigment through the agar. That's an identifying factor of Pseudomonas aeruginosa, which is ubiquitous. Ubiquitous. You spell that ubiquitous. Ubiquitous. And what it means is it's present everywhere. It's around us, on us, our clothing, the garments and so forth that we have on. It's just one of these organisms that's present everywhere. And we have a pretty good immune system, so as long as we have a good immune system, then we usually don't have any problem with Pseudomonas. It's a little gram-negative rod, and the rascal tends to be pretty resistant to antibiotics. That's right, Shavia, it's everywhere. Right here in the office with me. Now, you know it can contaminate burns because it's in the air. So one of the things they want to do, of course, is to cover the burns that where the tissue's been destroyed enough for the invasion of Pseudomonas to occur. As you look on page 592, come down to below the public health box. And you see Pseudomonas aeruginosa down there. And you see it says can be suspected when you have sort of a greenish discoloration at the burn site. Don't want to see that. Now notice the cultures, not the infection. Now, some of you may have worked in a hospital and maybe you've gone into a patient's room who's got a Pseudomonas infection. You know, it's got its own aroma. The aroma might be too nice a word for it. But you walk in there and you know exactly what's happening, just like a person with C. diff. Got that problem. It's got its own odor. But you see the culture can literally smell like grape bubble gum. So it's not offensive in terms of a culture. But anyway, it can create problems for us in terms of burns. Now, I also want you to look at the public health box right above that statement about the greenish discoloration. You see what goes around in whirlpool baths. whirlpools are nice, what they call them today. They call them jacuzzis and things like that. And it was sort of a big thing. I don't know if it is still, but everybody wanted their own jacuzzi on their deck or whatever. And the problem with jacuzzis is you have to clean them because people go in there and the oils on the body, the skin flakes and so forth. And, kids get in there and they don't want to go out and go to the bathroom, so they pee in there. And you end up with kind of an organic soup, which can encourage the growth of Pseudomonas in that jacuzzi. And if you don't take care of that and you get into that, it can create skin problems. If you look at the last paragraph under what goes round in whirlpool baths, that's under that public health box. See, they break out in a body rash because of the hypersensitivity reaction to being immersed in water filled with Pseudomonas. A swimming pool can be like that too, but they keep chlorinating the thing, don't they? That's the secret, evidently, to keeping that kind of a rash and maybe an infection from occurring. I had a student who did a seminar on that because he took care of the pool at McClinigan School, I think it was. They have a gym down there and a pool, and he had to make sure that the pool had a very low count of Pseudomonas. And so he actually recovered Pseudomonas from the pool water. Not much, only a few colonies, which was acceptable. We can't get rid of it. All we can do is keep it at a particular level, and then we can enjoy swimming in the pool. Our kids still get in there and pee because they don't want to go to the bathroom or whatever. It's amazing what we swim in, isn't it? Think about going in a lake. We used to swim in ponds all the time. All those critters in there. Obviously, we have a pretty good immune system. Everybody okay with the 2 Pseudomonas infections? Or the skin infections of Pseudomonas. There's only one, a burn infection, and then the irritation by the presence of a lot of them in a jacuzzi. Okay. So you look to the right, and you see in the next column where we're leaving the bacterial infections and we're talking about the viral infections. Now, what I want you to put beside viral infection, viral skin disease, I want you to put down fever blisters. Fever blisters, viral skin disease, caused by herpes simplex 1. Herpes simplex 1. Now, if you haven't had a fever blister, and I've never had one, I'm not sure why. Maybe I don't have a receptor for that virus. But my wife had them when we were young in our marriage, and she would get some look like sand traps. Boy, they were doozies and painful. So one of the things you want to realize is that they are very painful if you've never had one. Usually your immune system overcomes them. They have some products out now that shorten the lifetime, or the infection time, I should say. Instead of having it for a week, they'll have it for maybe 2 or 3 days. So they've got some things that work pretty well. Now, this virus, herpes simplex 1, is a latent virus. It hides within in the case of the fever blisters, it hides in the trigeminal ganglia that we have. They come from our brain. You have nerves that go to those ganglia. Got a number of different ganglia. So they are dormant in our case, usually one side or the other. They'll stay in that trigeminal ganglia until you have some sort of condition, a situation that will lower your immune response to some degree. It's not like you're going to develop AIDS or anything like that, but one of the things that seemed to trigger it for my wife was sunlight. So during the summer, when we'd go to the beach or something like that, within a few days she would have a fever blister on her lip. So it's still there, but she hasn't had any for several years now, a number of them. So she's very happy about that. Any question about herpes simplex 1 causing the fever blister? Come down to rubella. And you see it's also called German measles. Now note the description. We've had some measles outbreaks in America because people have refused to vaccinate their children. Now notice this, it says the mildest disease. Causes a skin rash, possibly. But it's usually about 3 days, 4 days, and it's not anything that's really bad. Sometimes people have it and they don't even know it. Now, you guys get vaccinated for that. So I've never been vaccinated for it. I guess I got rubella when I was a kid. I remember getting measles. And we'll talk about that in a few minutes. Measles, general measles is a lot tougher than rubella. But anyway, come down, let's see, from where you see mildest over there to the right. German measles is the mildest of several human viral diseases called exanthema. Come down 1, 2, 3, 4, 5, and you see often infected adult women often suffer from temporary arthritis and arthralgia. What we're concerned here, though, is you come down to the next paragraph and you see congenital rubella syndrome. So if you've got an infected woman who's pregnant, you see it says down in the next paragraph, it can transfer or it can cross the placenta and it can damage the developing fetus. What would that be called? Starts with a T. Back in chapter 10. Gosh, we only got 3 more minutes. You've been having as much fun as I have? I hope so. Anyway, think about that. See, that's a good question on the test. How does the rubella virus create birth defects? Think about it. Maybe you can mention it to me next Monday when we get together. Okay, it's 9:13, so we're going to knock it off. Do any of you have any questions? I don't know that I got all the answers, but you can surely ask the questions. That's right, Shavia. Teratogenesis. Now, think about this. How does the virus create that birth defect? So you want to be able to explain that. Then you'll understand why the birth defect comes into existence. Okay, well, I'm going to knock off here unless you have another question. I'll give you another 10 or 15 seconds if you want to ask a question. I'm going to be back on at 10 o'clock. I'm going to be in the lab and go through some things so that we can keep you updated on that. Looks like for at least for 2 weeks, we won't have a lab, but we can certainly get the information to you. Okay, Eugene says, what are we going to do about tests? Good question. I'm hearing that they're going to use a program called Respondus. And I think that program requires that you have a camera and maybe some other piece of equipment that you may be able to download so that I can put a test on D2L, and then you can open it up and you can take the test. You know, you've got these rules. That camera is going to be watching you, so you don't do anything wrong. I think you're not even allowed to leave the area or whatever. But you can take the test and then you hit the right button, and it comes back and I have to grade it. Until they set that up, we're dead in the water, so to speak. So as soon as they get it set up, I'll put a test that I was going to give you Monday on a D2L, and then we'll go from there. Okay, does that answer your question? Okay, guys. Well, I'll see you in lab in about 10 o'clock. Okay. All right. I'm going to end this. See you then. Okay. You're welcome, Eugene. See you in a few minutes.